Hey guys, it's Mrs. Kraus, and this is our third and final video on the Part 2 notes on meiosis. In the last video, we finished up by talking about possible ways that maternal and paternal chromosomes can line up along the metaphase plate during meiosis 1 to create different combinations of maternal and paternal chromosomes and also different combinations of genes in the final daughter cells. So the overall result of independent assortment in that particular parent cell we were looking at were 4 possible combinations of maternal and paternal chromosomes in the daughter cells. So some of the daughter cells had big A, big B, so both maternal chromosomes. Some of the daughter cells had little a, little b, so both paternal chromosomes. Some of the daughter cells had big A, the maternal A chromosome, and little b, the paternal B chromosome. And then some of them had the little a paternal chromosome and the big B maternal chromosome. Now we had these 4 possible combinations using the 2 to the nth power rule, where n stands for the number of chromosome pairs, and this cell had 2 homologous chromosome pairs, so 2 to the 2nd power is 4. Now these 4 possible combos of maternal and paternal chromosomes gave us these 4 possible gene combinations in the daughter cells. So some of them had blue-eye genes and straight-hair genes. Some of them had brown-eye genes and curly-hair genes. Some had blue-eye genes and curly-hair genes. And some had brown-eye genes and straight-hair genes. Okay. Now, both crossing over and independent assortment are often called processes of genetic recombination. And that's because in both crossing over and independent assortment, the genes on the chromosomes are recombined or mixed in different ways to give us genetically unique eggs and sperm. Right, remember, an egg cell and a sperm cell are going to join together during fertilization to create a diploid fertilized egg or zygote that has 46 chromosomes. So we see actually here real electron microscope pictures of a sperm fertilizing an egg cell. And this is actually a bunch of sperm trying to fertilize an egg cell, and only one of them will be successful. Now, eggs and sperm are both going to contribute the same amount of DNA to the zygote. They each contribute 23 chromosomes. However, the egg cell, if you'll notice, is much, much larger than the sperm cell. And that's because the egg contributes nearly all of the cytoplasm and organelles that are found in the zygote. All that the sperm really contributes is DNA. Now, the way that we can get this gigantic egg cell is by making cytokinesis happen unevenly in ovary cells. So meiosis in a human ovary cell is called oogenesis. It's not oogenesis, it's oogenesis. Okay. Now, we see in this picture, meiosis 1 happens to create our 2 daughter cells, and then meiosis 2 happens to give us 4 daughter cells. They'll all have the same amount of DNA. All of these 4 cells will end up with 23 unreplicated chromosomes. However, notice that cytokinesis is happening unevenly. So most of the cytoplasm and organelles from the original parent cell go into this daughter cell of meiosis 1. And then after meiosis 2, most of the cytoplasm and organelles from this cell go into this daughter cell, which is called the egg or ovum. If there are multiple eggs, they're called ova with an A. The other 3 cells, which are super tiny, are called polar bodies, and even though they have enough DNA, they've got 23 unreplicated chromosomes, they simply don't have enough cytoplasm and organelles to survive, so they disintegrate and die. But this ensures that we end up with one giant egg that is going to be perfect for creating a zygote, a fertilized egg. Now remember, the sperm don't need to be big. They only contribute really DNA. The egg's responsible for everything else. Meiosis in human testes cells is called spermatogenesis. Genesis meaning to create, so this is the creation of sperm. And cytokinesis that occurs in human testes cells during meiosis happens it should say the cytokinesis divides the cytoplasm evenly among the 4 daughter cells. So what we can see here in the image is meiosis 1 taking place, the daughter cells are the same size, and then meiosis 2 takes place, and the final sperm are all the same size, and that's because cytokinesis happened evenly. So we'll end up with 4 functional sperm cells that are all small. They're the same size, but it doesn't matter that they're small. Another question we can look at when we talk about meiosis is, do some people have more or less chromosomes than 46? And the answer to that is yes. There are some people that have different amounts of chromosomes in their cells. Typically, they would have either 45 or 47. When they have a different number of chromosomes than is normal, we call that aneuploidy. Aneuploidy happens when there's an error in meiosis that's called non-disjunction. So non-disjunction occurs when one of the following 2 events happens. So what we could have is a homologous chromosome pair not separating correctly during anaphase 1 of meiosis. So remember, during metaphase 1 of meiosis, the chromosomes line up in homologous pairs, and during anaphase 1, those homologous pairs split. Now, if one of the pairs doesn't split correctly, both of the chromosomes from that pair could end up in one of the daughter cells, and the other daughter cell would get nothing from that pair. Non-disjunction could also happen when the sister chromatids that are part of a single chromosome don't split correctly during anaphase 2 of meiosis. So here we see the chromosomes lining up single file along the metaphase plate like they do in metaphase 2. And this is anaphase 2, where we see the chromatids ripped apart, and once they're separated, they're called daughter chromosomes. They're unreplicated. Now, if those chromatids don't separate correctly, both of them could end up in one of the daughter cells, and the other one would get nothing from that chromosome. Okay. Now, this shows non-disjunction if a homologous chromosome pair doesn't separate correctly during anaphase 1. So both chromosomes from this pair could end up in one of the daughter cells of meiosis 1, and the other cell gets nothing. So if normal meiosis 2 takes place, both of the daughter cells on this side get one unreplicated chromosome from each of these maternal and paternal chromosomes. And they really should just have one of these rather than 2. So that those egg or sperm cells would end up with 24 chromosomes when they should have 23. On the right side, remember, we didn't have either chromosome from the pair. So even if meiosis 2 happens normally in the other chromosome pairs, we would still end up with one fewer chromosome, so 22 in the daughter cells when they should have 23. Now this is showing non-disjunction during anaphase 2 of meiosis. So we see that meiosis 1 happens correctly to put one of the homologous chromosomes in a pair into this daughter cell and the other homologous chromosome in the pair into this daughter cell. We see meiosis 2 happening normally over here where the chromatids split and we have one unreplicated daughter chromosome in each daughter cell. So if this happens correctly in all the pairs, we'd end up with 23 chromosomes in those daughter cells. However, on the right side, we see non-disjunction happening where both chromatids or daughter chromosomes end up in this daughter cell on the left. So that would have a total of 24 chromosomes, and neither chromatid ending up in this daughter cell. So it would have 22 chromosomes. Okay. Now let's say we made an egg cell that had the wrong number of chromosomes, it had 24, and it got fertilized by a normal sperm containing 23 chromosomes. You'd end up with a zygote or fertilized egg with 47 chromosomes, so the baby would end up having 47 chromosomes in each cell instead of 46. Now let's say the egg cell had 22 chromosomes instead of the normal 23. If it got fertilized by a normal sperm cell that had 23 chromosomes, you'd end up with a zygote or fertilized egg that had 45 chromosomes and a baby with 45 chromosomes in each cell. So we have some examples of real disorders that are caused by non-disjunction in humans. The first is Down syndrome, and that happens when the baby receives 3 chromosomes instead of 2 chromosomes for the 21st homologous chromosome pair. So we see that in this person's karyotype, which is a picture of all the homologous chromosome pairs in their cell, and we end up with 3 21st chromosomes when we should have 2. We should have 1 from the egg and 1 from the sperm, and in this case, we had 2 from either the egg or the sperm and 1 from either the egg or the sperm, for a total of 3. That means the baby has 47 chromosomes in each cell. Because it happens with the 21st chromosome pair, this is also called trisomy 21, tri for 3. This happens in about 1 in 700 births, and the chances of Down syndrome increase when the mother's over age 40 because non-disjunction tends to happen more frequently when the mother is older. People with Down syndrome do have a shorter average lifespan of about 35 years. They have common facial characteristics, as you can see with this little cutie right here, and they do have like low muscle tone as one of the symptoms. Patau syndrome occurs when there's trisomy at the 13th chromosome pair, so we see 3 chromosomes here instead of 2. Again, that baby will have 47 chromosomes, and it's also called trisomy 13. This is very rare. It happens in 1 in 10,000 births. The characteristics, people with Patau syndrome often have a cleft or divided lip and palate, so that's inside the mouth. They also may have an intellectual disability, and they often have polydactyly as well, which is where they have more than 5 fingers on their hands. Unfortunately, people with Patau syndrome typically die pretty young. They typically live about 3 months, and 80% die within the first year. Very sad. The next type of non-disjunction disorder is called Klinefelter syndrome, and this happens when there's non-disjunction in the 23rd chromosome pair, which is the sex chromosomes. So notice in this karyotype that the baby has 2 X chromosomes and a Y chromosome, when normally they would just have either XX or XY. Now this baby will be a boy because he has a Y chromosome. This baby is going to have 47 chromosomes in each of his cells. This happens in about 1 in 1,000 male births, and some of the characteristics of Klinefelter syndrome is underdeveloped testes, tend to be taller than the average male, and they may have some breast development. They are also sterile, which means they're unable to have their own children. This can happen if an egg that carries an X cell is fertilized by a sperm in which non-disjunction has occurred that has an X and a Y, or it can happen if there was non-disjunction in the egg and there were 2 X's in the egg. Last type of non-disjunction disorder that we're going to explore is called Turner syndrome. This is also non-disjunction in the 23rd chromosome pair, the sex chromosomes, and this is when the baby receives 1 sex chromosome instead of 2. It's called monosomy because mono means 1. So in this karyotype, we have 1 X chromosome and then nothing else. So we often represent this with an X and a 0. 0 indicates there's no other sex chromosome. So this person would be female because there's no Y chromosome, and she would have 45 chromosomes in each cell instead of 46. This happens in 1 in 2,000 births. Sometimes Turner syndrome isn't actually diagnosed until puberty, when the sex organs don't fully develop. Some other symptoms or characteristics you might see in someone with Turner syndrome would be a slightly webbed neck, so connected a little bit more to the shoulders, short stature, and then there are certain heart defects associated with Turner syndrome. Now, something that I think might be helpful for you guys if you are having trouble understanding the stages of meiosis, on the wiki page, underneath these videos, there is a McGraw-Hill meiosis animation, and I like it because, oh, is it not going to pull up? All right, well, it may not pull up in time, so I'll show it to you in class next time, and it really goes through the stages of meiosis quite well. Okay, so that is it for our Part 2 notes on meiosis. Give the notes questions a shot, and we'll answer any questions that you have next time.